1, 2, 3.
We only had 100.
Welcome to our next session of the bucket course. Can everybody hear me okay?
Yes.
Great. I am delighted to introduce to you Professor Emeritus of Biology, Bruce Voyles. Bruce received his PhD at Purdue and did postdoctoral research at the Michigan Cancer Center. And I could say lots of other things about him, but perhaps you read the article in the paper and know more about details. And some of you are undoubtedly colleagues of Bruce's at the college. Bruce and Martha moved to Grinnell in 1977. So did I. And raised 2 children here, Paul and Aaron. And Martha was a professor in the Department of Education while Bruce was in biology. He's going to speak to us about influenza A, a virologist's point of view. Let me give the usual 3 announcements at this point. Though I feel I often should go into the audience and just ask you to give these 3 announcements. 1, if you're using a T-coil, it's time to turn it on. 2, if you have a phone that hasn't been turned off or put on silence, please do that. And 3, we think we're going to try the system of people just raising your hands if you have questions along the way, and Professor Voyles will repeat those questions so that everybody can hear them and so that it works for the viewing audience, and we'll see how that goes. If you end up wanting the mic for some reason, I'm demonstrating the posture again. That's how it works. It doesn't work so well this way. It works this way. Welcome, Professor Voyles.
Thank you. I'm not used to this thing, so I don't. Is it loud and clear? Not quite right. Okay, that sounds better. I would like to do this somewhat informally. I've been told that I'm not supposed to ask you questions. And unfortunately, that was my teaching style. So this is going to be kind of awkward for me. Hence, everything is going to be on the PowerPoint. I'm not holding anything back in order to frighten you by asking a question. I always on my end-of-course evaluations, I got these things that I was so intimidating, and I never have figured out in 30-plus years of teaching how I could be intimidating. I'm just the nicest guy in the world. If you prepared for class. And fortunately, you don't have to prepare. Let me give you a brief outline of what I'm going to do today and next week. And that is, I'm going to start by attempting to scare the bejesus out of you by talking about the 1918 pandemic flu. Then I'm going to introduce you to viruses. I'm sure everybody in this room knows what a virus is. I'm willing to bet 95% of you don't know what a virus is. So I'm going to tell you and introduce you to a few of my very tiny friends. Then I'll talk specifically about influenza A virus. And I'll talk about it from a variety of perspectives, its structure, its reproduction. A very important topic there will be how it changes year to year, so that you always need to get a flu shot, a point to which I shall return often. Although I know you've all been good and gotten your flu shots already. And then how it changes to become a pandemic virus. That's one of the aspects of the virus that is unique. Its mechanism of gradual changes or large changes. And then finally, depending on how much time we've got, at the end, I'll talk about how prepared we are now, what we have by way of antiviral drugs, and they're constantly being developed. So perhaps by the time the next pandemic comes, and it will, we will be better prepared. I will also talk about vaccines and why you need a new vaccine every year and why they're not terribly effective. But they're better than nothing. So, get them anyway. They will help you some, perhaps. And you can just think of it as your government in action. I probably shouldn't say that around a bunch of people from Mayflower and college faculty and stuff. I grew up in Idaho, which is 70% owned by the government. And so they were always regarded as the enemy. And I'm afraid I have retained some of that worldview. Anyway, it has nothing to do with being a virologist. Let me see. Is that right? Okay. So, part one, the 1918 pandemic. And I do want you to ask questions, but as you can see, I'm not very tall, and so I may miss you in the back, in which case just yell out, and Janet will help me get the question, okay? So the 1918 pandemic. The pandemic, whoops, I see it. All right. Oh, well, this is fun. Managed to get the first slide wrong. Anyway, what's on the missing slide is that in America, one of the first recorded cases of the 1918 flu was at an Army base in Kansas, where they were bringing in recruits from all over the Midwest, and there were about 50,000 young men at a time in this base, and they gave them basic training and tried to sort of feed them up to be stronger. They were apparently a pretty pitiful bunch in terms of their general health. But in March, specifically March 18th, 1918, a new recruit reported to the infirmary, and he was very sick. And very shortly after he appeared, another and another, and then hordes of them. And the whole base was affected. They didn't all have the flu at the same time, but they all were certainly exposed to it. As you would expect during wartime, these recruits were sent to Europe whether they were healthy or not. And they were sent to the East Coast, put on ships, sent to Europe, and from there to the battlefields. Well, of course, you couldn't have devised a better way to spread the pandemic than putting these sick people on very crowded ships and lots of time to spread it. And then, of course, when they got to Europe, they dispersed and spread it widely rather. The symptoms of the 1918 flu made it very clear that it was something new. Not just the fact that it spread very rapidly, but the symptomology was quite different from ordinary flu. They had what was called a rib-cracking cough. In other words, coughing so hard that you felt like you'd done damage to yourself. You had also accompanying that a high fever and dark spots on the cheeks were also evidence that you had caught the flu. After you had the flu well established, the next sign that something was terribly wrong was that your fingertips and ears and so forth began to turn blue. And that's called cyanosis. And it was because you were not getting enough oxygen. And the reason you were not getting enough oxygen is that your lungs were filling up with a frothy, bloody substance that you would spit up or cough up. And it was evidence of massive pulmonary bleeding. And you would die as a result of not being able to breathe. So it was a very unpleasant illness and certainly not what we would call a good death. And I'll talk later about the symptoms of what's called the seasonal flu, the one that comes through every year, and you'll see how different this was. Now, I tried to do a lot of research on numbers and discovered that you could pick any number you wanted practically as how many people were infected, how many people died, and so forth. And I figured, well, I might as well give you the most grisly numbers that I came across. And we really have no idea how many people were infected and how many people died. For one thing, even in countries like the United States, where people were given death certificates, at that time flu was not a disease that was reported to the public health officials. It was not a reportable disease, so the doctors could put down any number of vague words, and there was difficult to interpret them. So even in countries like the United States, we don't have especially good numbers. And in countries like China, for example, it's anybody's guess, essentially. So it was estimated that 20 to 30%, well, a third of the world's population was infected. And of those, that third, 20 to 30% of people who had the flu died. And the estimates range anywhere from 30 million up to 100 million people died as a result of this flu. And that's died in a very short time window, not like other diseases where it may be a longer period of time to get a mortality figure. If we take the 100 million, that's more people than died in the military if you combine World War I, World War II, and the Korean War. So it's a very significant loss. And it was 3 to 6% of the world's population that died from this illness. Let's see, come on. I attempted to put this in figures that are relevant today. As of August, the world's population was somewhere like 7.7 billion people. In 1918, it was about a seventh of that. If 1/3 of the world's population now became infected, it would be 2.6 billion people. And if 20 to 30% of those died, it would be between 513 and 619 million deaths. That's pretty serious stuff. And it is why periodically, as I'll talk about, it appears that the same flu virus is coming back. And needless to say, that alarms people a great deal. Turns out that it hasn't come back yet. But in some respects, that doesn't matter because there are also other viruses that can cause this problem. Now, I'm going to show you on this slide and the next slide one of the things that I always discuss with my students, and that's the presentation of data so that you do not give the wrong impression. Now, if we look at this slide, it says 29 million deaths in the 5th month of the pandemic, and all the red areas are where deaths occurred. Now, I am going to ask a question. I'm sorry, I can't. What's wrong with this slide? If we're talking about 29 million deaths all over the world.
It doesn't even have China in it.
Yeah, it's Eurocentric. It doesn't include half the world. And those data were, available. This is what the world looked like. All the red areas were where there were significant deaths. And so the first slide was probably not deliberately dishonest. I think they probably just only had room to do the first half. But nevertheless, always be be an observant viewer. Okay. You may have seen some of these pictures, especially this one. This shows an influenza ward. As you can see, it's a very large room, completely filled with beds. And I'm not certain, but I think this was at a military base. This one is to illustrate the effect that the flu pandemic had on society in general. People came to realize rather quickly that the more closely packed together people were, the easier it was for the virus to spread. And so places where the public would assemble, like theaters, were closed. And probably at least some of you remember in the 1950s when the polio epidemic was going along. And it was the same thing. I can remember theaters closed, and this was in the summer. The theater, air-conditioned theater, which was the only place that was air-conditioned, was closed. The city swimming pool was closed, and so it made for a rather miserable summer. But the really miserable part was the palpable fear, especially on mothers, I think. And the same thing was happening here. Not only did they close places like theaters, mayors went so far as to urge churches not to have services. That went over real well, as you can imagine. They also asked businesses to stagger the starting and quitting time for employees so that everybody was not piled onto the public transport system at the same time, getting to work and coming home from work. So they were trying very hard to reduce large population clusters, to reduce the spread of the disease. Here are a group of women who are carrying stretchers, and behind them are black painted vehicles, and the women are dressed in black. So those are clues that these women are not nurses. Instead, they pick up the dead and cart them away. And there were so many deaths that cities couldn't keep up with burying them. And so this is a picture of digging mass graves in Philadelphia. It reminds me of the Black Death in Europe. But it's evidence that society is beginning to break down, that there are so many deaths that basic services, if you will, like burying the dead, are not possible in the usual way. Okay, so what caused the flu? Now, in 1918, the germ theory of disease was well accepted, and that is that bacteria caused infectious diseases. The disease causing bacteria you could see in a microscope, and they could be grown outside the body on artificial media, okay? And that's how you identify a causative agent of the disease. A bacterium was isolated from many flu patients when they were coming to the end and spitting up bloody sputum. And so this organism was called Haemophilus, blood-loving, influenzae, of loving, okay? So this blood-loving influenza-causing organism was thought to be the causative agent. Unfortunately, it wasn't. It was simply what we call an opportunist, an organism that takes advantage of a particular environment in order to grow. And many of us have Haemophilus in our respiratory system, and it causes no harm. So prior to the development of germ theory, for thousands of years, diseases were thought to be caused by miasma, a vapor that spread in the air. Related to that was the use of the word virus. And virus is a poison, and its use generally was for airborne poisons, okay, the miasma. And the only thing that was known about viruses in 1918 was that they were something that was small enough to pass through the pores of a filter that would retain bacteria. So these were small somethings, smaller than bacteria. All right, so any questions before I go on? Okay.
I have a
Yeah, Mari?
You said they're expecting that virus to come back, so it must mean it's hanging around someplace? And where would that be?
I think it's in the park. No. Yeah, the question was, I said that the 1918 virus is hanging around, and I will qualify that a little bit later. The particular strain of virus that caused the pandemic is currently a pandemic virus. It's sort of reappeared, but it's not exactly the same virus. It's pandemic, but it is not causing the 1918 flu, okay? And that will become clearer when I've talked about how we identify the pandemic viruses. There have been efforts, however, to locate actual 1918 flu viruses, and they have gotten some out of, one was from a woman, an Inuit woman who was buried in the ice, and so they got some from her tissues. Was there, no?
Bruce, this is a semi-statement and a partial question. You don't usually see this in the history books, but I think, and would you agree, that the end of World War I, the armistice, is partly due, if not mainly due, to this pandemic? It's hitting the armies in the trenches and so on. They lose the will to go on because they're fighting something that is insidious and destroying them without them destroying each other.
Yeah. What George said was that World War I ended in part because all of the armies involved in the conflict were suffering significant mortality from the influenza. Again, I saw estimates as high as a third of all the military were sick, and a significant number of them died. So it did have apparently a very major effect on the soldiers in World War I. I was about to say, one of the things that gets me into trouble is I say things to be funny that probably aren't. But I was about to say, I guess there's one thing we can be thankful for with this virus, but anyway, probably not true. Okay, yes?
You mentioned that the first case in the U.S. was maybe in Kansas, in the military. Is the original source of the epidemic known?
The original source is not known. Many people think that, like lots of other flu viruses, it originated in Asia. And again, I'll talk about that in more detail in a little bit. Somebody somewhere in the larger Midwest managed to get infected. You know, maybe went to San Diego to visit his aunt or something, and came back and then went into the camp because that was a collecting point for a large radius, and so undoubtedly he was not the first person to die of the virus, but he's one of the first where it's certain, with the Army then as now loves to keep records of things, so he was documented.
How does a pandemic like this burn itself out without infecting the whole world?
Well, the question is, how does this burn out without infecting the whole world? And the answer is that we talk about the 1918 pandemic, and it was actually the middle of 3 cycles of the virus going around the world. And it's the one that was the most significant. The first and third did not cause anywhere near the deaths that middle one did. And so by the time the virus has gone around the world 3 times, probably most people have at least encountered it. They didn't necessarily get really sick, but their immune system caused them to be resistant to it. And as an aside, if I had my immunological kit with me, I could tell you your age within a short bracket of time, because obviously the older you are, the more pandemic viruses you've encountered, and they're one every 10, 15 years, and so I can put you in slots according to which viruses you have an immune response to. Anyway, this is not a lab class, so okay. Part 2 is, what is a virus? Now, I'm assuming that you all know the word virus, and you all know that people get sick because they get a virus and so forth. But I'm willing to bet if I ask you to describe one, what it looks like, how it works, you know, maybe, I don't know, Professor Bubbles might be able to give some answer, but, I think I saw, yeah, I think it's possible a few of you could have an answer, but most of you really don't know what a virus is. So I hope you will indulge me, or you can sneak out. I want to talk at some length about what a virus is. So what characterizes a virus? Well, I already mentioned it's small, but that's not a distinguishing characteristic. There are bacteria that are as small as viruses, and there are viruses that we're discovering primarily out of seawater that are larger than a fair number of bacteria. So small, it's not a totally distinguishing characteristic. Viruses can't be grown on artificial media, but neither can the vast majority of bacteria be grown on artificial media. It's mostly the bacteria that are of interest for causing disease that we've worked out a way to grow them in culture. A third characteristic may be mentioned, and that's that these are obligate intracellular parasites. Obligate means they have to be taken in by some cell, and they grow then inside that cell, if you will. So they're obligate intracellular parasites. But so are the bacteria in the groups Rickettsia and Chlamydia. And so again, that's not a distinguishing characteristic. So what does characterize a virus? The major significant difference is that a virus exists in two totally different forms. There is an extracellular form that's called the virion, and I'm not going to ask you to remember very many terms, but virion is one I use frequently. The virion contains only proteins and nucleic acid. And one thing that distinguishes the virion from a cell is that the virion contains only one type of nucleic acid. All our cells contain both DNA and RNA. Viruses contain either DNA or RNA, but not both. Okay? And, well, the RNA viruses are a huge group, and to my mind, very interesting. Okay. The nucleic acid, either RNA or DNA, is surrounded and protected by a protein shell, and we call this the capsid, and it's directly taken from the Latin word for box. And it is a protein shell that protects the nucleic acid, the genetic material of the virus, from environmental factors which would degrade it. Now, and that is a virion, this capsid plus nucleic acid. And it's what you can image in an electron microscope. And I'll be showing you pictures of lots of different kinds of capsids. In addition to the capsid, many viruses have a cellular membrane surrounding the capsid. We call this, for once in a term that makes a lot of sense, an envelope. And the virus is, the virion is the letter that is going inside the envelope. And this is a cellular membrane, something the virus picks up. Now, Janet is making motions. Right? Look at the last thing. I have this all by itself. The virion is dead. It has no metabolic activity whatsoever. And that's what distinguishes live from dead in cells. If a cell's alive, it has at least some form of metabolism keeping it going. If it doesn't, it's dead. Okay? And this is what leads to this metaphysical discussion about whether viruses are dead or alive, because they're certainly dead, in quotes, when they are in the form of a virion. We'll see after the break that when they're in the cell, they're very much alive. And so, unless you, I don't know, some strange kind of resurrection occurs when it goes into a host cell, okay? But I used to tell my students, you know, if you want to know if it's dead or alive, go to the philosophy department. Okay.
That's a great thought to end on. Really, for most questions, we should go to the philosophy department. We're going to take a 10-minute break and listen for the bell.
Okay. What I was described before the break was the extracellular form of the virus, the virion. A major distinguishing feature between the viruses and cells is that the virion disappears in all or part in an infected host cell. Usually the situation is that if you examine an infected cell, the cell will show evidence of being infected. It will swell up, it will get big holes in it, or it'll get foamy looking, and you can look at it and say, ah, there's a sick cell. But you won't see anything that looks like a virus. It's gone. Okay? Some viruses leave some remnant that will be visible, but most of the time, the intracellular form of the virus is the nucleic acid of the virus. It doesn't need the envelope, it doesn't need the capsid, it's just the DNA or RNA that's important. And it's important because the virus subverts its host cell metabolism to make viral nucleic acids and proteins. It takes over the host cell and stops the host cell from making its own proteins and its own nucleic acids, so that it can use all of the precursors of those large molecules to make virus proteins and DNA or RNA. So it's a genetic parasite, if you will. Okay, now, a cardinal distinction between cells and viruses. Host cells, in fact, all cells, divide to make daughter cells. The dictum is all cells from cells. Ever since the very first primordial cell in the whatever sludge back 4.5 billion years ago came into existence, it must have divided to make 2 cells, and they divided to make 4, and so on. 4.5 billion years later, here we are. And the only way that our cells can make new cells is to divide. New virions are not created by division. They are literally assembled from pools of their constituent parts. There's no division involved. They make the nucleic acids, they make the proteins, and they fit the proteins around the nucleic acid, and then at the end of the creation of new viruses, they may pick up cellular membrane as they leave the cell. But it has been demonstrated with a number of bacterial viruses that you can build a virus in a test tube. That if you obtain all the constituent parts, and the experiments that do this, have each one in its own tube, and if you add them in the right order, the virus assembles, and it will infect host cells and so forth. You can't do that with cells, although people are trying. Okay, now what I want to do for the next few minutes is show you what a bunch of viruses look like, and I will show you electron photomicrographs, and then what we call cartoons. Anyway, they're not funny, they're simply illustrations of what we want to describe. Now, on the left side are the virions of tobacco mosaic virus. This is a false color image. The electron microscope does not take pictures in color, simply black and white. But for stock photos like this one, I got all these off the web. They frequently colorize them because it makes it easier for people to see what the image is. Now, tobacco mosaic virus is an RNA virus, and it has a very simple architecture, as you can see. The RNA is covered with a capsid that is simply protein units that have a groove in them. So the RNA will fit in the groove, and it just goes around and around and around and creates this stick-like virion. Tobacco mosaic virus is the first virus to be shown in the electron microscope. And so it's not just its simplicity, but its historical significance that makes it the first one that we always show. So this is what is called a naked helical virus. Naked in the sense that it does not have an envelope. Not that it's naughty. The other form of capsid is based on the icosahedron, a 20-sided geometric solid. And we often call these spherical viruses. And the small ones are very clearly icosahedral. As they get bigger and bigger, they become more spherical, as you'll see. But this is, oops, back up. Okay, this is parvovirus, which is in this case, a dog virus, and you may have gotten your dogs immunized against this virus. As you can see here, I hope, these are icosahedral, and this is a cartoon of what this looks like, and here is a drawing, and here you can see the nucleic acid, and it is surrounded by capsid proteins that form an icosahedron. Okay? Now, a little bit bigger spherical virus are two closely related viruses, poliovirus and rhinovirus. Rhinovirus, you know what a rhinoceros, is the long-nosed dinosaur. Well, rhinoviruses are cold viruses, and poliovirus is poliovirus. In the electron photomicrograph, the regularity of the capsid is quite evident because they crystallize, they fit together, and that's why I chose this one. You can't really see the individual ones well, but you can certainly see that they fit together. And this is what the virion looks like. It's got points that are surrounded by 5 identical proteins, and then it has other places surrounded by alternating proteins, and there are 6 of them. And this 5-6 pattern is repeated now as the virions grow bigger and bigger. Okay? I tossed this in just because the viruses have everything imaginable in terms of architecture. This particular virus has 2 capsids, one inside the other. And this is one of the viruses that you can actually see a part of when it's in the cell. The outer capsid is removed, and the inner capsid remains as a structural entity, and the viral nucleic acid is synthesized inside that inner capsid and poked out of the holes in the capsid. And there are wonderful photomicrographs of the capsid with RNA molecules going all over the place. But anyway, so, and this is an animal virus. Plant viruses have a whole bunch of different organizations that involve 2 capsids, or dividing the genome up into 2 or 3 different capsids, all sorts of things. Anyway, now the envelope viruses. And this is one of the larger ones. This is an enveloped helical virus, and it's the rabies virus. And it has a very characteristic architecture. There are these bullet-shaped virions, and sorry, I'm getting klutzier as the morning goes on. The viral genome starts with a small loop and then gets larger until it reaches this sort of constant size, and then it stops. And so the membrane is then taken up around this, and it produces these bullet-shaped virions. And for a number of reasons, I really like rabies virus. Anyway, we won't go there. This one I don't like. Enveloped helical virus, and this is mumps virus. And I think most of us in this room had mumps before the vaccine became available. I really feel like my children missed out on a lot of character-building events by being vaccinated against everything. It's a little hard to see, but if you use your imagination, perhaps you can see these kind of stripes, and if you were to look more closely, you'll see that they are coiled up nucleic acids. And the virion is spherical, and I liked this particular figure because it shows you that the nucleic acid has attached to it the enzymes that will make new copies of it. So it's all carried in a package, and then it is surrounded by an envelope, and envelopes consist of what are called matrix proteins, which line the inside of a membrane at a position where the virus will leave the cell. And so the membrane is sort of an identifying mark for the virus as it leaves the cell, and then it picks up this outer membrane, and it's usually modified, that membrane, by putting what we call spikes in it. This is one you've probably seen. It's Ebola. And Ebola has a very interesting architecture. It's this flexible, chain, and it has the nucleic acid inside and then a very close-fitting envelope around it. I, over the years, have spent a lot of time saying, don't worry about Ebola. You know, so 25 people died of it over in Uganda or whatever. Don't worry about it. It's not coming here. Unfortunately, this virus appears to be beginning to escape and has much more widespread occurrence in Africa, and is kind of worrisome now. I wouldn't be so cavalier with my class anymore. Okay. Another envelope spherical virus is Herpes Simplex. And this is a more complicated virus. As you can see, it has an icosahedral capsid inside of an envelope, but between the envelope and the capsid are another group of proteins, and so the virus has this very complicated architecture, and these proteins are called tegument proteins, and they're involved in synthesis of new nucleic acids and then the assembly of the virus. And then another enveloped helical virus that does not display either rod shape or spherical. This is HIV. Has a very characteristic virion, and the envelope surrounds this sort of truncated pyramid shape of capsid. And so it's essentially unique, very easy to identify. This particular virus and others of its group, the retroviruses, have 2 copies of their genome. It's an RNA genome. And when this virus infects a cell, it first makes a copy of this genome in the form of double-stranded DNA. So it takes the RNA and makes a DNA copy of it. And the process is remarkably complex and involves starting on one of these strands and then at some point hopping over to the other and doing some more stuff and hopping back to the first one and so forth. But it gets the job done. Then the DNA form of the virus is integrated into the host cell's chromosome. And we have many, not HIV, but we have many retroviruses, which this is called retro because it's going backwards. It's going from RNA to DNA. But our genome is full of these, and they don't cause diseases or whatever. They are evidence that the retroviruses have been around for a long time, and we pick them up periodically. And so they show our ancestry, certainly back to the beginning of the mammals. Okay. Questions before I move on to the actual topic of this talk? 15 minutes before we have to end. Gene?
As a chemist, I'm amazed by the self-assembly that goes on. Can you comment on that?
Yeah, it is amazing.
Can you repeat the question?
Oh, the question was, he says he's amazed by the self-assembly process. And how does that go on? Well, each unit type has a structure such that it will bind to another unit, either identical to itself or different. And so the self-assembly is based entirely on protein nucleic acid or protein-protein interactions. There's nothing that from the outside that builds the nucleocapsid and the other capsid. And in fact, there is a virus that's a little bit larger than polio virus, but somebody has isolated all the intermediates in the building of the capsid and showing that it starts with making the 5 protein unit, and then it hooks the 6 protein units around that, and then the 5 units come in again, and so forth. And again, you do it entirely in the test tube. So, you know, there's some good, I think, structural chemistry there. And it's the same with these, I should say, with these naked viruses, like polio virus is a naked icosahedral virus. And it shows 3 different proteins on its surface. But it has a 4th protein that is inside, locked to the surface proteins of the capsid. And it's locked so that dynamic equilibrium, I think, is the term. If not, I just made it up. Sounds good anyway. But what happens when the polio virus attaches to its host cell surface is that there is a change in the shape of those surface proteins of the capsid, and that 4th protein is allowed sort of out, and it opens up the capsid so that the genome can get out. And so again, that's a very, I think these sorts of things are very clever of the virus, but it's got a mechanism to open up the capsid by having this protein in a sort of, the inside protein in a sort of stressed environment. Okay, part 3, we'll get a start on this. I'm told that I absolutely positively have to shut up at 11:30. Anyway, so now we're finally at the end of the first session, getting to influenza A virus. Here is a colorized electron photomicrograph. You can see that the virus is surrounded by an envelope, and that envelope looks kind of fuzzy, and it's because the envelope is covered with spike proteins, proteins that are sticking out from the envelope. And so it has this characteristic appearance of the spikes. This is some artist's imagination of what it looks like. It does not look like this, obviously, but I did want to show you that there are 2 different kinds of spikes, okay? The first one of these spikes is called the hemagglutinin molecule, and we abbreviate it H for, sometimes HA for hemagglutinin antigen. Heme is blood. Agglutination means clumping. So this is a blood clumping spike. Now, hemagglutination is a way of identifying viruses and quantifying them in the laboratory. Any virus that attempts to infect a red blood cell has just committed suicide, because red blood cells are just sacs. They don't have any of the metabolic constituents or activities that the virus would need to reproduce. And so this is sort of an unfortunate name, but it's ubiquitous. Everybody's got hemagglutinin spikes, seems like. But just bear in mind, it's not really talking about an infection. Now, the one I want you to remember when you go home, is the neuraminidase spike. And neuraminidase cleaves N-acetyl-neuraminic acid from sugar chains on the surface of host cells. And that sugar always occurs at the end of this sugar chain. And so neuraminidase is cutting off the last sugar of this chain. And for shorthand, I don't know, but I can't remember now what sialic acid is. This when it's at home, okay? But I can't remember what the derivation of sialic is. Okay. This, again, is showing the virus with its spikes. And I should have pointed out before this, the electron microscope, like all microscopes, sees a section of the object that's under the microscope, in this case, viruses. What you see here are these circles with the ring of spikes around them, and that's because the beam is focused partway through the virion. Okay, and I'm sorry I didn't point that out sooner. This does not show you the three-dimensional virus. You just see cross sections through it. The other side here is a cartoon of this. And again, it shows you that there are two different kinds of spikes. There are the hemagglutinin spikes, which are the, what color that is, beige, gray, anyway. I apologize to the artists in the audience. And the orange are the neuraminidase spikes, and there are a lot fewer of them. I want to point out, and I hope you can see this. Here's a neuraminidase, or a hemagglutinin spike. Here is another one, and so forth. And this center one, you can see, has three identical components. So we call a trimer a 3-unit molecule. Okay? And that's important for how the virus gets in. Okay, as I mentioned, we don't infect red blood cells. The hemagglutinin spike attaches to N-acetyl-neuramic acid, and sugars, and these are 6 carbon molecules in this case. Sugars can combine with each other or be linked to each other through at least 3, possibly 4 of the different carbons in their molecule. And each way that something's attached, I have my thumbs together, then one thumb stays the same, and this is a different sugar and a different sugar, and so forth. So a carbohydrate chain doesn't have to be very long to be essentially unique because of all the permutations. And so N-acetylneurominic acid occurs on the surface of all kinds of cells, but the conformation that is identified by the hemagglutinin spike occurs only on the cells of the respiratory tract. Okay? And we say that this establishes the host range. For influenza A, has to have this particular configuration of the last sugar and the penultimate one. Okay? And that reminds me to tell you that there's no such thing as stomach flu. All right? You've got a rotavirus or something else, but it's not influenza. Influenza is of the respiratory tract. Okay? So if you want to say stomach flu, go stomach flu. Okay? Anyway, and if I hear you say it, I'll correct you. It's one of my pet peeves. All right, there are 16 different types of hemagglutination spikes. And this 16 becomes important. Well, all right, before I do it, don't worry about this. I'm not going to ask you to reproduce it on the quiz next time. But this is what a hemagglutinin spike looks like. And I told you it's a trimer. And, whoops, if you look at the colors, you can see there's the blue-green, the pink, and then behind it is a white one. So 3 of them kind of swirl around each other, and then at the top, there's a sort of ball-shaped structure. And here are the 3 different balls, if you will, that come together to make the spike. Okay. Am I done? Or do I?
After this, let's say you're done after this.
Okay. The other spikes, neuraminidase, play their role at the opposite end of the virus's life cycle. The hemagglutinin spikes help identify a proper host cell and get the virus into that cell. The other end of the process is getting out of the cell. And neuraminidase, as the name, cuts neurominic acid off of the cell surface so that the H spikes can't bind to it as the virus is leaving. Otherwise, it got in with the H spikes, it tries to get out and gets trapped, and that's no good. So it's got the neuraminidase spikes to allow it to get out. There are 9 different kinds of neuraminidase spikes, 16 different kinds of hemagglutinin spikes, and that means there are 144 possible combinations. So we are not going to run out of different kinds of flu viruses anytime soon. Okay? So with that, I think that's a pretty good place to stop.
Thank you so much. That was very interesting.
Yeah, thank you.
And we look forward to hearing about part 2 next week.
Yeah.